Hi, hello everyone. Welcome to our channel Insta Notes. Come join with us and learn with us. Today's lecture is all about a very important cell organelle, the mitochondria. So, mitochondria is especially present in both plants and animal cells. So, this is a eukaryotic cell showing the presence of mitochondria in it. So, what does the word mitochondria mean? Mitos means thread and chondrion means granules. The mitochondria is present in the form of thread-like granules all scattered around the cell. So it is usually present in the cytoplasm of plants and animal cells. It is generally called as the powerhouse of the cell. Why? Because the mitochondria is the site of energy production for the cell. The energy is produced in the form of ATP, that is adenosine triphosphate. So let's look upon the outer structure of mitochondria. This organelle is a double membrane organelle. The outer membrane is more permeable and contains forins, whereas the inner membrane has two compartments, the outer and the inner matrix. So let's see what happens in the matrix region. So cristae. The cristae are the convolutions or infoldings of the inner walls of the mitochondria. This cristae causes increase in surface area so as to perform different functions of the mitochondrial matrix. So let's dig deep into the matrix and look upon what happens exactly in the cristae region. So the enlarged cristae here shows pin-like studded structures around its membrane. They are called as the Stark particles. Let's further zoom in and look out what are these Stark particles. They are nothing but the oxysomes. These oxysomes are very important for oxidative phosphorylation reactions. As its structure denotes, it has a head region, base region, and the stalk. So oxysomes are the sites of oxidative phosphorylation. In the head region, it has F1 particle. In the base region, it consists of F0 particle. Before going on to the reactions of oxidative phosphorylation, we'll look upon some of the basic elementary particles of oxysomes. They are the F0 F1 particles, the electron transport particles, inner membrane subunits, and the Fernandez Morin particles. So, coming on to oxidative phosphorylation reactions, it is the process of ATP formation when electrons are transferred by electron carriers such as NADH or FADH2 to oxygen. So let's look upon what happens inside the matrix region. So the protons, that is H plus ions, pass through an enzyme called ATP synthase, which is present in the head region of the oxysome. So this H plus ions pass through the oxysomes and enters the matrix region, where the ATP synthase complex helps in the generation of ATP from ADP and inorganic phosphate. Thus, the cell's energy is produced. So let's look upon the individual functions of the membranes of the mitochondria. The outer membrane contains transport proteins for shuttling pyruvate, which is the end product of glycolysis, into the mitochondria. This is the intermembrane space between the outer and the inner membrane of the mitochondria. The matrix region has appropriate enzymes and suitable pH for the Krebs cycle reactions. As I said before, cristae is highly folded so as to increase the surface area and volume ratio of the mitochondria. The inner membrane especially carries out the electron transport system and the ATP synthase for oxidative phosphorylation reactions. So, coming on to the key facts about this mitochondria, which will be very helpful in terms of examinations. So, the mitochondria is first observed by Kolliker, and the term is coined by Carl Bender. So, these are the two scientists we have to remember in terms of mitochondria. The size of mitochondria is zero point 0.5 to 2 micrometer in diameter and 3 to 4 micrometer in length. Coming on to the composition of this organelle, it consists of 70% of protein, 25 to 30% of lipids, 5 to 7% of DNA, consisting of 70S ribosomal unit. The presence of DNA makes this organelle to be named as semi-autonomous organelle. The single circular DNA molecule is usually present in the matrix region of the mitochondria. This mitochondria is inherited only from a female parent. It multiplies by fission reactions. As I said before, it is present in the plants and animal cells, whereas mitochondria is absent in prokaryotes. There, the mesosome perform the function of mitochondria. So, how we could observe mitochondria under a microscope? So, the vital strain used to observe mitochondria in live cells is Janus green B. It is also called as a pleomorphic organelle since it shows variable in shape. It is usually not found in prokaryotes and mature red blood corpuscles. The number of mitochondria is more in more metabolically active cells. So this lecture will be followed by few questions which usually repeated in the competitive examinations. So the mitochondrion is enclosed by, yes, it is double membrane layer. The enfoldings in the inner membrane of mitochondria is called as, yes, it is the cristae and not the cisternae. Cisternae is present in the Golgi bodies, which we will deal with in the future lectures. Mitochondria are the sites of, yes, they are the site for both aerobic respiration or oxidative phosphorylation. So the answer is both A and C. So in this lecture, we have seen about the oxidative phosphorylation reaction and ATP generation. And in future lectures, we will deal with aerobic respiration that happens in the mitochondria. Which of the following is called as the semi-autonomous organelle? Yes, it is the mitochondria. Yet another organelle which is considered to be semi-autonomous organelle is the chloroplast. So mitochondria and chloroplast. Mitochondria consist of, yes, since it has a single circular DNA, it has 70S ribosomal unit in it. In mitochondria, most of the enzymes for electron transport system are present in, yes, it is in the inner membrane of the mitochondria. All enzymes of Krebs cycle are found in, yes, it is in the mitochondrial matrix. Mitochondria replicate by, yes, it is not the fission reaction, it is the strand displacement model. It multiplies by fission, whereas it replicates by strand displacement model, which is the site of Krebs cycle in bacteria. Since mitochondria is absent in prokaryotes, that is bacteria, mesosomes perform the function of mitochondria. So the answer is mesosomes. Mitochondria are usually found in, yes, both A and B. It is present in reproductive cell as well as vegetative cell. So if you have listened to our lecture very carefully, you can answer all these questions very easily. So thank you for listening to us. Thank you.